Thanks for checking out the RSA lecture series. I'm Sara Jo Nixon, and in this presentation, I'll be talking about the neurocognitive consequences often seen in alcohol use disorder. First, let me describe some of the characteristics of the individuals that we'll be discussing. When individuals who have been chronically consuming excessive amounts of alcohol initiate abstinence or detoxification, they may experience a period of acute withdrawal accompanied by a host of physical and neurological consequences, including irritability, attention problems, cognitive issues, seizures, DTs or delirium tremens, and even death. In short, alcohol withdrawal is a medical event and should be undertaken with medical supervision. Underappreciated is the fact that even for individuals who don't experience that sort of acute withdrawal episode, that there are a number of residual cognitive consequences. According to the Diagnostic and Statistical Manual of Mental Disorders, version 5, 30 to 40% of persons with alcohol use disorder demonstrate clinically significant levels of compromise. Empirical studies also show that 50 to 80% of individuals studied exhibit significantly poorer cognitive performance than their community comparison groups, who are equivalent in terms of age and education. Finally, carefully controlled studies have shown that these neurocognitive deficits cannot be attributed to comorbid conditions that also affect cognitive performance. More specifically, with few exceptions in the studies described in this presentation, we'll be referring to men and women who meet criteria for moderate to severe alcohol use disorder according to current guidelines, or alcohol dependence under the previous DSM-4, and they are all engaged in treatment. They will have completed detoxification if it was required. They are not taking significant psychoactive medications, such as antipsychotics. They are generally healthy and are typically evaluated about 21 days or 3 weeks after their last use. This period is generally assumed to account for a period of subclinical withdrawal. Much of the early work derived from the field of neuropsychology, a field of study directed to understanding the links between compromise in specific brain regions, initially defined by major external landmarks, such as those demarcating the cerebral lobes, and internal landmarks, such as major white matter pathways, and tying these areas of the brain to cognitive and behavioral compromise. While the field of cognitive neuroscience has evolved, neuropsychology remains a core component of its exploration. The brain areas and their interconnections may be seen as modulating or mediating broad functional domains. For example, executive functions, which are associated with frontal and prefrontal areas and their connections, reflect decision-making, inhibitory abilities, and activities, such as planning and problem solving. The left temporal lobe is critical to verbal learning and verbal memory, while the right plays a significant role in visual spatial processing. Perceptual motor skills engage the somatosensory and primary motor cortex, which lie on either side of the central sulcus, while language reception and production engage the temporal lobes, the temporal parietal junction, and aspects of the frontal cortices. These rather broad localizations may guide empirical study and facilitate interpretation, but cognition is not easily compartmentalized and is clearly dependent on the intrinsic connectivity among these brain areas. This slide illustrates neuropsychological tests that probe key functional domains. In the upper left-hand corner, you will see the block design The block design requires that individuals align these 3D black and white cubes to align with the 2D picture and places significant demands on visual spatial processes. The picture on the upper right shows the grooved pegboard. In this task, individuals pick up each peg and it has a notch which they turn and must place in each of the pegs. They have to do so in a particular order. It is timed, and they have to do so with both their dominant and non-dominant hand, testing perceptual motor skills. In the bottom left, you see the digit symbol substitution test. Here, individuals must precisely copy the symbol, placing it beneath the appropriate digit. They cannot skip a digit and must proceed in a specific order. The DSST certainly has a psychomotor and visual scanning aspect, but it also engages executive functions. Finally, in the bottom right hand, you'll see an example of the verbal fluency or oral word fluency test. Here, individuals are asked to produce as many words as they can that begin with one of the letters, with some restrictions, such as not repeating a word or using a proper noun. There are also versions in which individuals must generate examples of semantic categories, such as animals and vegetables, and again with some restrictions to not repeat themselves. Oral and written fluency is sometimes administered in written form, integrates multiple skills, but is particularly dependent on executive functions. In the next couple of slides, I want to demonstrate two of these tasks because we'll be showing their sensitivity to alcohol use disorder subsequently. Here is the DSST, and as I mentioned, you are not allowed to skip a digit and must proceed from left to right. This is the only two of many lines, and individuals have but 90 seconds to complete as many as they can. I also want to note the specificity and precision with which these symbols must be made. As you can see, this looks as though it is a U, but it has squared sides, and that makes a difference in the scoring. Here is the trail making test, Trails B. Trails B forces set shifting between numbers and letters, unlike the Trails A, which is all about following the numbers. Thus, while it has a motor component, it also requires set shifting or executive functions. Individuals are timed for their completion, and errors are accounted for by the increased time that's required in order to make up for that. We and others have used the DSST in a number of studies of alcohol-related cognitive dysfunction. This slide shows recent work in which community residing and treatment-seeking men and women were compared. As you can see, the AUD group completed significantly fewer symbols than did the control group, again reflecting compromise in executive function, as well as likely involvement of visual scanning and reproduction deficits. In this slide, we show the Trails B. I mentioned that just a moment ago. And again, we see an alcohol-related deficit in completion time for the Trails B, with alcoholic men and women taking significantly more time to complete the task than did the control men and women. I picked these examples because they tax the executive functions, and executive functions rely heavily on the integrity and integration of frontal and prefrontal systems and are therefore essential and critical to effective daily conduct. Furthermore, they are commonly compromised with alcohol use disorder. There's been continuing attention to the question of whether the right or the left hemisphere are more or less sensitive to alcohol's negative effects. By manipulating the task parameters, such as the nature of the stimuli, it is possible to make this comparison. This slide, although depicting earlier findings, is a wonderful example of such work. Here, Sullivan and colleagues compared community controls and persons meeting criteria for alcohol use disorder on a variety of tasks where they were pointed to the right hemisphere by using things like pictures and visual spatial stimuli rather than words. Here, even though the domain names are a bit different than what we talked about earlier, you will notice that except in grip strength over here and in design fluency, there is significant compromise in right hemisphere functions, whether we're looking at executive function, short-term memory, declarative memory, and aspects of motor ability. When stimuli tasking the left hemisphere were employed, the impairment was less uniform, as you can see by the changing blue lines here, but still significant deficits were seen in aspects of executive function, declarative memory, some aspects of word recall, and finger movement. Thus, let's put these together. So we've got tests of right hemisphere and tests of the left hemisphere. If we lay them on top of each other, as you can see here in the Lateralized test composites, there's a contrast between the left hemisphere, that is the orange lines, and the right hemisphere and their relative performance to age-corrected standardized scores, or Z-scores. Notice that really, except for perhaps declarative memory in the left hemisphere, there are significant deficits in both hemispheric functions across these various domains. So it isn't just that one hemisphere or the other is more or less sensitive. And I want to bring up something else. Notice that we talked about the importance of the frontal lobes and their interconnections. These exist, of course, for both right and left hemisphere domains, and they are compromised whether you're looking at executive functions mediated in the right hemisphere or the left. This pattern, reinforced by a large body of work, albeit incompletely consistent, demonstrates the generalized mild brain dysfunction that characterizes cognitive function after detoxification. The study of brain electrophysiology using non-invasive measurement from scalp electrodes has also been an important tool in ascertaining alcohol's long-term effects. Neurophysiology is exquisitely qualified to clarify the temporal dynamics of neural activity and provides assessment of brain activity at both rest and during task conditions where shifts in response to specific stimuli or responses can be ascertained. These shifts are referred to as event-related potentials. A commonly studied event-related potential is the P3. The P3 is a positive shift in the brain's neural activity according to about 250 to 450 milliseconds after the presentation of a relatively novel but meaningful target stimulus. For example, in an oddball task, a person might see a green dot 80% of the time and a red dot 20% of the time, and be asked to keep track mentally of the red dots. In that case, the red dots elicit a larger P3 amplitude than do the green dots. Note this amplitude is typically dampened in alcohol use disorder. This dampening has been interpreted as reflecting an impairment in contextual or memory updating. Alcohol-related deficits are also seen in other ERPs, presumably linked to more complex cognitive abilities. So let me show you that in this slide. This slide shows the brain's electrical response to semantic congruity or incongruity. Note that in the style of traditional electrophysiologists, negative is graphed upwards. Individuals are shown a series of sentences one word at a time. In some cases, the ending is consistent with the rest of the sentence. In others, it's related, but not consistent. And finally, in others, it is inconsistent and highly irregular. For example, in the sentence, the pizza was too hot too, the expected word would be eat. And when eat is presented, there is really very little shift in a negative direction. When drink is used instead of eat, you get a shift in the N400, but certainly not as big as that associated with the word cry. And when cry is presented, you have a significant shift in the N400, that is about 400 milliseconds after the presentation of this final word, suggesting the detection of semantic incongruity. We were able to use this task in a large sample of alcoholics and community controls, in which we examined the related incongruent response, that is like the drink response, and the unrelated, such as the cry. When we did so, we found that for the related incongruent, drink and eat, there were very few differences between the alcoholics or those with AUD and the community controls. However, when we examined differences in the unrelated incongruent, we found that, let's just focus on this square, that the AUD group had a significantly dampened amplitude to the incongruency of that semantic ending. This is important. Why? Because it suggests that when things don't quite make sense, there's less of a neural response to that confusion than there is in community controls. And when you think about this in the real world, you can anticipate that there may be confusions in communication, a lack of certainty in interpretation, and a need to perhaps clarify what it is that one might mean. And indeed, that brings us to the next phase. So far, we've talked about performance on laboratory tasks, and these tasks are essential in characterizing alcohol-related impairment. But we recognize that the problems that drive individuals to seek treatment do not commonly arise from poor performance on such laboratory tasks, but rather problems in navigating daily living. We've had the opportunity to look at some of these issues, and this next task is an example. This is the plant task. And this task comes from a Piagetian framework. In this task, you actually show individuals each of these plants. 2 look healthy, 2 do not look healthy. They are not healthy because they have either been dried in the sun, or if they were stolen, they have been placed in the microwave so that they look a little wilted. For the first plant, you say, taking care of this plant, how does it look? Looks pretty good, doesn't it? Well, every day I'm giving this plant some blue plant food and a small glass of water. You then move to the second plant. Again, the first situation remains in front of the participant and say, this second plant doesn't look very good, does it? Every day I'm giving it yellow plant food and a small glass of water, but I'm also using leaf lotion. You then turn to the third one, repeat the fact that this plant looks pretty good, and describe the fact that you're giving this plant blue plant food using some leaf lotion, and although it's not obvious in this slide, I apologize, a large glass of water. And finally, you go to the fourth pathetic plant and say, every day I'm giving it yellow plant food and a large glass of water. You move then to a side table where you say, I have another plant that I'm taking care of, and every day I am giving it blue plant food and a small glass of water. And you ask 2 questions. Because it's Piagetian, one of the questions is, how will my plant do? Good, bad, 50-50 chance. And then you say, how do you know that? So that this task asks individuals to not only identify, but isolate the relevant variable inside the problem. When we did this and looked at the data, and these are question number 2. So we asked question number 2, how do you know that? Only of those individuals who guessed that the plant would do fine. You all have already figured out, no doubt, that the answer has to do with the blue plant food, not the size of the water, not the leaf lotion, etc. And what we found was that the AUD group, which based on the age of this article was labeled ETOH, they perform much more poorly. They were able to get the same number correct in terms of how's my plant going to do. In fact, they outperformed the controls. But when we then say, how do you know that? We find that the individuals with AUD were less able to isolate the blue plant food and instead combine things with the blue plant food instead. Question 4 is an ancillary question that is not really relevant to our current discussion. Unless you're a florist, plant task might not be the most important thing in the world, but one of the things we absolutely know is that interpersonal problems are a hallmark of individuals seeking help for substance use disorder and for many others of us. Using an interesting battery of vignettes, we examined the typical and best responses offered by those who were in treatment and community controls. Here's an example of the vignettes. The topics varied from relationships with partners or spouses to children to work environments, and even in some vignettes related to alcohol use. We took the alcohol vignettes out because we knew that would bias responses between the community controls and the individuals with alcohol use disorder. So here is one that is an example of the workplace, and actually it could be any 2 friends who are sharing information. Later, you discovered that what you said about the acquaintance has gotten back to that person. You feel really embarrassed and hurt. What do you do? There are scoring guides that direct scoring to the degree to which violence, alcohol use, hateful emails are issued, and more productive approaches are provided. We also ask individuals 2 questions. In the first round, we ask them to tell us what their typical response would be. And in the second, we ask, what do you believe the best response would be? In doing so, what we found was those with AUD performed significantly more poorly than the controls in terms of the quality of their typical responses, that is what they would typically do, but not in their anticipation of what the best response actually was. It was hypothesized that perhaps this was due to differences in self-efficacy, but as you can see down here on the right, that could not account for these differences. I might note that the significant sex effect in the best condition was due to women providing better best responses. This is an old paper, obviously. We had the opportunity to revisit this task in more recent work and were delighted in terms of assessing the reproducibility of the outcomes and perhaps the validity of the task, and found again that there was no difference between groups or sex in terms of best. In the previous one, we found a sex effect for best responses. We did not get that here. But we, more importantly, found that the AUD group was inferior in terms of the quality of their typical responses. That leads nicely to the next issue of interpersonal challenges. The ASB is one way of doing that. You're asking about hypothetical, though realistic conditions. And for decades, however, scientists and clinicians have recognized the importance of emotion processing in AUD. As noted here, there are a number of studies illustrating alcohol-related impairment in emotion identification, but almost none integrating study of emotion and traditional aspects of cognition. So with support from NIAAA, we've had the opportunity to probe this interface. In the study I'll be describing now, we use the emotional face expression task, and in it, we morph emotion along a continuum between neutral to 100%. We only use the 35, 65, and 95 for study. And what we had individuals do were make judgments of 3 emotions: anger, happiness, and sadness. So in each case, they had to pick whether that face was happy or sad or angry or sad. And for this task, it's really important to have a face processing control task. In that, we used the sex judgment task, in which we took pictures of men and women and blended them. Some of these worked better than others along a continuum. And in this case, asked individuals to judge, let me go back one, whether or not a picture was male or female. So in both cases, faces were being judged and shown, but they were being judged and shown on different characteristics. What did we find? Well, we found that the alcohol group was indeed lower, had lower accuracy across all morphs, whether they were 65 or 95. Let me say here that we ended up taking out the 35% because it was so difficult for all participants, regardless of group. So we found that the AUD group had lower accuracy across the morphs, that is, whether it's 65 or 95, and the emotions. The greatest difference was at the 65% angry, and the second at the 95% angry. This played back to something that's been supposed in the literature, and that is that negative emotion, and perhaps particularly anger, may be differentially difficult for individuals with AUD to deal with and approach. Beyond group differences, we wanted to examine the relationship between emotion identification and interpersonal problems. There was a significant relationship, but only for the AUD group and only for anger, calling back again to the significance of anger processing deficits in AUD. Interesting, the correlation was significant for the subscales of needy, non-assertive, and overly accommodating on the interpersonal problems inventory. Some propose that it might be related instead to those that are more externalizing, but what we found were these issues with anger processing were related to problems in being not assertive enough and having problems with being able to accommodate or set boundaries. Here, we used the emotion processing test that I just described and looked at emotion processing by these groups. Here's the emotion processing. But in this task, we also had other measures of cognitive function, such as the TRAILS-B that we've talked about before. And here again, we have the alcohol-related deficit. The DSST, again, we have the alcohol-related deficit. And now we have the emotion judgment accuracy. When we pulled the sex factor out of the work that I just talked about, we found that the emotion judgment deficit was largely driven by the alcohol women, not the men, suggesting again the sensitivity of sex differences, and particularly the role of sex differences and emotion processing in the consideration of alcohol use disorder. Across all emotions and morph levels, the P3 amplitude that we obtained by look at each of the faces, the P3 amplitude elicited in response to the EFEs was significantly smaller among AUDs relative to the community controls. And it was whether it was happy, sad, or angry. In the lower right figure, you see again in a different format the mean amplitudes. By here we're showing the 35% as well as the 65 and 95%. So these deficits in the P3 were not specific to a particular morph level or a particular emotion. Again, suggesting a more generalized deficit in terms of the contextual updating that may be what the P3 reflects. So where to now? What we do know is that there are broad generalized deficits. They can be observed across diverse processes and in tasks ranging from well-characterized neuropsychological tests all the way to impersonal and interpersonal problem solving and emotion processes. What we want to know is, can we determine potential mechanisms that underlie these diverse deficits? And might they aid in determining interventional targets? We and others are focusing on the possibility that examining cognitive control processes will shed light on this issue. While it's difficult to draw direct parallels between laboratory tasks and those with greater ecological validity, we have proposed that a common deficit may indeed lie in the operation of cognitive control. Cognitive control engages top-down processes to facilitate intentional goal-directed behavior. There are several models, but one that we are particularly interested in is the conflict monitoring model. And it is characterized by both an evaluative and a regulatory component. In the evaluative component, the individual is facing the issue of, what is the stimulus? What was my response? Was that a congruent stimulus? Was that an incongruent stimulus? Did I make an error? And in the regulatory component, this information is combined so that effective behavioral adaptation occurs across trials. Enhancing the ability to identify stimuli appropriately, such as in the emotion task, and or mentally register mistakes may enhance longer-term outcomes. Similarly, effective behavioral change over time and in the face of changing stimulus conditions is the essential characteristic of psychosocial adaptation. We are indeed delighted that some initial pilot work from a cognitive training protocol, that these data are quite promising, and we hope to be following up on those in the next years. In summary, a robust literature reveals generalized cognitive disruption in individuals with AUD, even after detoxification. Importantly, these deficits are seen not only in well-characterized laboratory tasks, but also in tasks with greater ecological validity and those interrogating emotional processing. Necessarily, we have constrained discussion of individual differences variable in this lecture. These factors we know may contribute to the heterogeneity we see in alcohol studies. We have also not discussed recovery functions. These are important issues that merit separate consideration. However, it is also important that we mention that these deficits improve with time. Although measurable disruptions may exist for months and in some cases even years, these recovery trajectories occur. They are influenced by sustained abstinence, but much is unknown regarding individual differences in these improvements or if specific training intervention might facilitate outcomes. These latter points give reason for hope, and it is imperative that individuals with AUD, their families, and friends are aware of and embrace such change. Finally, before I close, I want to thank you for being a part of this series. I also want to thank a bevy of collaborators, the support from NIH and primarily NIAAA, and others, as well as private foundations. And especially, I want to thank the participants in these studies who have contributed so much to our understanding. Thank you for listening.